Guys, so we are going to be working on part 4 of our notes today, which is going to look at genetic engineering. It should be pretty quick. There's not a lot that we need to cover, just about, I don't know, 10, 12, 15 slides, something like that. Anyway, it's genetic engineering, so you should have your part 4 notes up. It starts on slide 21 of our slideshow, and we shall begin. And that's it for this unit. So let's go. All right, human activities that increase genetic variation. So there are several ways that we can actually make species or populations a little bit more genetically diverse. The first thing, and we do this all the time, is selective breeding. And that's where we choose organisms with wanted characteristics to produce the next generation. We specifically do that when we're looking at dog breeds, cat breeds, anytime you're breeding fish or birds, any animals that we breed for specific things, or even if you look at like dairy cows can be bred to the ones that produce more milk are the ones that get reproduced more often so that they're passing those genes on. Okay, so that's selective breeding. Hybridization is when you cross dissimilar individuals to bring together the best of both. And you see this with Labradoodles. They were taking Labradors and Poodles, and the Poodles had no shedding, and Labradors have an excellent temperament, and they bred them together, they hybridized them, and they made the Labradoodle. So that's how we made a new breed of dog, is through hybridization. We can do that with plants as well. So if you've ever had a pluot, that is a hybridization of a apricot and a plum, and they bred them together to cross them together to get that new fruit called the pluot. Sometimes we have to introduce mutations through either radiation or chemicals, and that can cause mutation rates to change, which will bring about variation. Not always a good thing, and I'm not sure how well controlled that is, but that's definitely a way that we can have mutations occurring. If you look at, or if you think back to the Chernobyl and Fukushima guided reading that we did, that was radiation caused chemical, or radiation caused mutations to occur, and some of those mutations were good, some probably not, but either which way, you now have more mutations because of those introductions of radiation. Also, another one is polyploidy plants. It's a tongue twister. That's when you have drugs that prevent the separation of chromosomes during meiosis, and we talked about polyploidy in I think part 1 or 2, and it was that instead of having one go in each gamete, or 2 wind up in each gamete during meiosis 1, you would have 3 in one and 1 in one, and that would not be correct. You would have one too many chromosomes in one and not enough in another. Okay. All right, so now let's talk about some recombinant DNA, and that is DNA that is containing multiple sources. So you have some from maybe bacteria, and then you have other organisms, and then it gets combined. Specifically, we use plasmids, which are small circular bits of DNA that we get from bacteria and yeast, and we can use that to make recombinant DNA, to make DNA do what we need it to do. So first you copy DNA from a source, and then you change it by introducing this other DNA source. Okay, so let's look at genetic engineering, and normally we would be doing a plasmid lab where we make a paper plasmid, but unfortunately this year we will not be doing that, and it really helps you understand that you're actually cutting out bits of DNA and putting in new bits, and it's definitely a unique process, and it's definitely one that's helped with things like creating bacteria, or not bacteria, antibiotics and other therapeutics. Okay. So genetic engineering is basically the process of manipulating genes for purposes that we need. Like, there's all sorts of purposes. We do it for food products. We do it for animals. We modify things inside them to, maybe for cows, for instance, to produce more milk. Okay. There's 4 main steps, and you have to identify 4 main steps. The first is to cut the DNA, and to cut DNA, in the past when DNA was cut, or kind of cut open is what we talked about for replication. of DNA and mRNA for transcription. That was DNA polymerase coming in and RNA polymerase coming in. So that was an enzyme that broke apart those hydrogen bonds. This time, instead of cutting apart the bonds, we're cutting it into smaller segments. And there's, instead of DNA polymerase or RNA polymerase, we're looking at restriction enzymes that come in and do that. And they make smaller sections that can then be used for making recombinant DNA. Then you have to clone them. You want to get as many copies of that as you can or that you need for whatever you're going to do, whatever the purpose of your process is. So you cut it, you make your recombinant DNA, then you clone it, so you get lots of copies of it, and then you have to screen it. And cells that have received the particular gene are screened from the ones that didn't to make sure that it's in there correctly and that there's not any issues happening with that. Okay? So cutting, making recombinant DNA, cloning, and then screening are our 4 main steps. Okay? One of the processes that we can do to find out if the genetic engineering has worked is a gel electrophoresis. This is one thing that gel electrophoresis is used for. It can be used for other things as well. But that tests, you can use it to test for the presence of the engineered gene. Your DNA fragments are cut into different sizes and they get separated by size. So how fast they move across the gel electrophoresis plate, which is charged with electrodes so that it will attract the DNA to move down it. And you'll have smaller chunks, the smaller fragments of DNA will go faster and farther down the plate. So like this is positively charged and your DNA fragments will move towards the positive end. So if it's smaller, it'll go even further. If it's a bigger piece of DNA, a bigger segment, it will stay close to where we input the DNA, which is in these wells down here. If you want to practice doing this at school, you can take, I believe it's forensics, and they will have you do gel electrophoresis, or I believe in AP Bio, you will also get a chance to do this. I actually got to do it in college. I didn't get to do it in high school, so it's kind of cool if you get a chance to do that. All right, genetically modified crops are also called GM crops or GMO. And oftentimes we'll hear of non-GMO things because there's a lot of genetically modified crops is a very controversial topic. At times, science is wishy-washy on whether it's necessarily the best thing, but overall I think it's found to be fairly benign to do what we've been doing, and most of us wind up eating genetically modified food without even knowing it on a regular basis. So gene technology can be used to create plants which can tolerate drought, which is really important if you look at California's climate changing to being more dry and having more frequent droughts. If we can develop plants that can withstand that and still produce the fruit and veggies that we need to get from it, the yield, then that's probably a good thing for at least the human population. You can also have them to tolerate different soils. Maybe some prefer clay soil and others prefer sandy soil, but if you don't have the right soil where you are, maybe you can get a genetically modified crop that can handle the soil that you have in your yard or your fields. And also climate changing is definitely something that we would want to be able to have plants withstand, because we don't want them to die if there's droughts or poor air quality. Maybe we can have some that have a higher photosynthetic rate when they can take more carbon dioxide out of the air. I don't know if that's a possibility or not, but it's something to think about. Okay, so why might genetically modified crops be beneficial or harmful? And I'd like for you to take a minute and pause this right here and write down what you think. Why do you think genetically modified crops would be beneficial or harmful? And maybe you have some strong opinions. Maybe you have done some background research on this already. If you haven't, just based on what we just talked about, you're probably going to have a very simplistic idea of why it may or may not be beneficial or harmful. Normally we'd have a whole discussion and bring up everything in class, but again, this isn't typical. So just based on what you know and what you just heard, do you think it's a beneficial thing or a harmful thing? You have a couple of lines there. Go ahead and fill that in and then restart the video as soon as you have finished doing that. All right, moving on to genetically modified animals. And I did talk about, maybe cows producing more milk earlier. We also have transgenic animals, and these ones have human genes added to farm animals. This is kind of cool. It's different. I'm not sure if it's necessarily the greatest thing ever, but it's pretty cool that we've been able to genetically modify cows to produce milk with human protein in it. That's then great because then we can take that and then extract it from the milk and sell them to the pharmacies can sell it for, and they can sell it to the pharmacies to make different things for them and test on them and everything. So it's got a lot of pharmaceutical purposes. So we want to make sure that we can take advantage of those types of things if we can because it's helpful. Okay? All right. And this is how it happens. Next, health and medicines. Sometimes, just like we're in the middle of our pandemic here, vaccines are useful to help prevent diseases. Genetic engineering is 100% useful in helping to develop vaccines. We saw this firsthand this year, how we took mRNA of COVID-19 and we were able to create this benign vaccine that can help prevent people from getting it. It gives you the response, the immune response, but then doesn't get you the actual virus, which is super important, especially this year when we're talking so much about getting vaccines and all that. If we did not have genetic engineering and the technology that we have now, we would not have the vaccine that we have, and that would be a very sad situation that we would be in. So we got to thank our lucky stars that we have technology and understanding of mRNA and genetic engineering to create these really awesome tools in our belts to fighting things like COVID-19. Also, we've been able to use transgenic animals as test subjects to simulate human diseases for medical purposes. So we can simulate which therapies might work if we can get the same response out of animals if they have everything in place, like the human proteins or whatever it might be. So it's kind of a neat way that we do that. And then we can also use recombinant bacteria can produce human proteins or be used for gene therapy, which if you are watching this before Tuesday's video, we're going to watch a video on Tuesday, Wednesday about gene therapy for sickle cell. But if you're watching it after, then you already saw the video on sickle cell and how gene therapy may very well be used for that and how we can modify stem cells, our own stem cells, to produce the correct blood cells if you have sickle cell. It's just one technology that's being implemented to help treat and maybe eradicate. I don't know if it would eradicate it because I'm not sure if that person would then pass on the corrected genes or if that person would still, I don't know how that all works, pass on the sickle cell. So that's interesting. I would actually like to look that up. So gene therapy is huge and genetic engineering makes gene therapy possible. Okay. Basically, you'll put a healthy copy of a gene into stem cells of a person whose copy of the gene is defective. So the one that we're watching, if you have seen it already, was the sickle cells, and they took the stem cells from the girl and inserted the correct gene, and then she made, in the course of 9 months, all of her sickle cells went away, which is really amazing, and she's not having the side effects of it anymore, of having sickle cell. So it's phenomenal, and I really am anxious to see where this technology will lead us. All right, next, DNA fingerprinting. And this is, again, something that we use for, and this is why you will do it in forensics. It's used in forensics to find and identify people's genetic DNA markers, and you can identify suspects. And we talked a little bit about this with, what did we talk about that with? They talked about the gal on the 2020 episode who found the Golden State Killer through using pedigrees. That's right. And they had the DNA analysis of different individuals on a pedigree, but they didn't, and then they had to fill in the pedigree to find it. So all of this is actually quite connected when you start looking at it in a bigger picture. So if you are interested ever in becoming a forensic scientist, you could be using gel electrophoresis and pedigrees to help find suspects, which is kind of cool that you're learning stuff that will help you down the road. Anyway, let me get back to this. DNA fingerprinting is an individual's DNA creates a unique pattern of fragments when separated by gel electrophoresis. So these are the bands, and remember, the farther away you go, the smaller and faster those fragments have moved. And gel electrophoresis, another definition for it, is a technique for separating molecules according to their size. Okay. These patterns can be used to identify persons. So if you have a matching set, then you can say, okay, those two are identical versus this suspect does not match the latter specimen. Okay, so that's how you can figure it out. We also use it for paternity cases, besides forensics, also in identification of deceased individuals. We can get samples of hair follicles from hairbrushes or toothbrushes and identify and see how those samples compare. And they're also using it in wildlife conservation to see which species DNA samples they're collecting and to kind of get an idea of how large the population size is, if it's endangered, if conservation efforts are working, if there's too much inbreeding going on, which we talked about with the Isle Royale project, or I guess it wasn't a project. When we did Isle Royale, we talked about how much inbreeding was going on, and how that one lone wolf was able to diversify it enough at that moment. But we can tell all of that from doing these DNA gel electrophoresis analysis. Cloning. So we're going to do talk about some cloning as well. In 1997, the first successful cloning was done, and it was a sheep named Dolly who was produced. I happened to be in high school in biology class in 1997 when this happened, and my teacher gave us a copy of the article that came out in, I want to say like Newsweek or something like that, about how amazing this was, that this animal survived and lived and they were able to do this. It was mind-blowing when it first happened. Like, oh my gosh, imagine the possibilities of this. Dolly was an exact copy of the donor sheep's DNA, and we will, if you haven't watched, if you're doing this before Thursday, Friday's lesson, we're going to watch a whole episode or 23-minute clip on 60 Minutes on cloning of polo horses, which is being done now, and you have entire teams of a cloned horse down in South America. And so not only is cloning of animals here and today being done, it's being done very successfully, but it does also lead us to some huge ethical and moral concerns about whether human cloning it should be allowed. And there was in 2004, there was a Chinese, or no, South Korean, they said they successfully did a human embryo and they cloned it, but then the results they later found out was a fraud. They didn't actually finish cloning it. So that's definitely something that is up for debate as to whether or not we should even be messing with humans and cloning them. But the other part of it is not necessarily cloning humans, but can we clone organs? Because we have a major shortage of organs. Can we clone healthy livers and pancreases and lungs and hearts and kidneys? I mean, if that is something that we could look at doing, and I've also heard of not just cloning, but 3D printing the clones. Is that something that we're willing to do? So maybe we're not willing to do a whole human, but maybe we're willing to do parts and harvest parts for medical purposes to help. Maybe it's your own parts. I don't even know. There's so much out there that we don't know yet, but it's definitely an area that I think you will find a lot more information coming down the pipes throughout the next decade or so as we get better at 3D printing and as we get better at with our cloning technology and as we understand the consequences of doing it. I don't know, it'll be interesting to see if that's something that our society ever grows to accept. So I think that's it. That's it. Have a great rest of your day. Please bring any questions you have to class. Hopefully you watched this before your videos. If not, then I've just previewed all the videos for you for nothing, and you've already seen the videos and it all makes sense. But otherwise, if you are watching this before the videos, sorry, I did preview them for you. You'll enjoy them, I think. So I do enjoy watching the clips and stuff. So we're going to talk a little bit more this week about sickle cell and cloning and wrap up our units on Thursday, Friday. So have a great rest of your day. Hopefully you're doing this before spring break. And I will see everybody after spring break. Have a great break.